Hi, Kristen. Can you hear everybody? Or can you hear me? I hope this works.
Yeah, I can.
Yeah.
It wasn't working this morning, but I think everything is good now. All right. Okay, so we're on 8.3 today. And oh, you can't see. Okay, let me get this turned on. So 8.3, we're going to learn about DNA replication and how DNA is copied in a cell. And once that happens, the cell then divides into two or more cells. And so remember we talked about mitosis and meiosis before. So before those things happen, you need DNA to be duplicated through replication. And once it's duplicated, then you can make egg cells, sperm cells, or you can make more of the same kind of cells, depending upon what your body needs. DNA replication copies genetic material of a cell. It copies the entire DNA strand. It takes about 8 hours for this to happen, which is really incredible if you think about it, because you have 3 billion nucleotides. And that, again, that's like copying 1,000 books, 1,000 pages long. That's the equivalent of doing that. Your body can do that in 8 hours. And one reason why it can do it so quickly is because there are about 100 different places it's copying the DNA strand. If it didn't do that, it would take about 33 days to copy one DNA strand. But because there are over 100 different places that are being copied simultaneously in the DNA strand, it can copy the whole thing in about 8 hours. And one DNA strand in your cell is about 6 feet long when you unravel it. And so imagine 6 feet information, but it's billions, 3 billion nucleotides of information within there. So replication copies genetic information and makes a new strand of DNA. A single strand of DNA serves as a template for a new strand. So a template means a model or a blueprint. Remember we learned about the different stages of the cell cycle, G1, S, G2, M. So M is mitosis, where the DNA or the cells are divided into two new cells. G1, the cell is growing. S, that's when the DNA is duplicated. So we're going to focus on the S stage today and learn about how DNA is duplicated during that stage. So as DNA is copied, the base pairing rules apply. So you have an enzyme that goes down the DNA strand and it looks at the nucleotides. Let's say it looks at A. If it sees A, it's going to pull in T. If it sees C, it'll pull in G. So it's going to do the mirror image of what it sees on the DNA strand. And that's what they mean by the base pairing rules apply. Whatever you see on one strand, the complementary component to it is then pulled in to build the new DNA strand. DNA is replicated during the S stage, synthesis. And each new body cell gets a complete set of DNA, an exact copy. So every cell in your body has the same exact DNA. And it's done through mitosis. Yeah, Lucy?
So why does every single cell in your body need the full set of DNA?
That's a good question. Each cell does use a good amount of it. I wouldn't say a small part of it. The other thing is that your cells can then turn off and turn on genes. And so it's able to control what genes are turned on in a certain part of the body. And that's what makes, your heart cells, your muscle cells, your liver cells. based on its location, it can turn off and turn on genes. And that's a lot easier to control it that way than trying to cut out genes. So that's what would have to happen. If you have, different cells, you could do two things. You could turn off that gene, or you could just try to cut out the gene you don't need. That's would be two ways to do it. Because when the first cell of the baby is formed, that cell has a complete set of DNA. And so when the new cells are formed, you could have two options. You could cut some of the DNA out, or you could just turn off the DNA. Your body decides to turn off the DNA in certain places. It's a lot safer to do it that way than to cut DNA, because you can make mistakes when you cut the DNA. You can have mutations.
Also, does each chromosome have a different set of DNA, or is it just different?
I tell you, like 1, 2, 3, 4, 5 to 23.
Yeah.
Like, yes, so each chromosome has a different kind of, different patterns of DNA to make the different genes. That's right. Because each chromosome is different. It has its own set of genes. Did you all get this down? Hey, Hannah?
So is like one gene or allele just a pair of A and T? Or C and G?
Usually a gene is about 100 to 1,000, sometimes even more nucleotides. It can be below 100, but generally, it depends on the kind of gene you're talking about. But usually in the hundreds is what you're talking about as far as how many nucleotides make up a gene,
I see.
at least.
And so how does like a gene work? Is it just a chemical reaction?
Yeah, so it's a code to make proteins. And those proteins then give you a certain trait. So hair color, for example, if the genes have a certain code that makes a certain protein, and that protein gives you a certain hair color. If it makes you melanin, you get dark hair. If it makes pheomelanin, you're gonna get red hair or blonde hair. So based on the kind of protein you make, you get a different hair color. Skin color, same thing. Melanin is what makes your skin dark. So a certain gene makes a lot of melanin, you'll get dark skin. So it's controlled through proteins. By making proteins, you then get certain traits.
I see.
So it's hard to imagine how all our traits are controlled by proteins, but they are. Proteins are very versatile. Proteins can make machine-like things in our body that do certain things. Proteins are chemicals that can break down or build up things. They can have certain colors. And so by making certain proteins, you get all the traits that we know that come from our genes. So proteins also carry out replication. So there's certain proteins that pull apart the DNA. I mentioned the other day that, DNA normally is double helix right over here where it's intertwined. But when it needs to be copied, it has to be pulled apart. And so hydrogen bonding, we talked about this, that it has to be pretty weak to easily pull apart the two DNA strands. And when it's being pulled apart, it can now be copied. So during replication, you see this happen, where the DNA is pulled, each strand on different sides, so it can copy one strand. And proteins are what pull the DNA apart, and a different protein then comes along the strand and copies the DNA. And DNA is a template that tells it what to do. So if this red is an A, then it's going to then build a T on the other side right here. If this is a C, it's going to bring in a G on the other side. And it'll keep on doing that down the row. And this gold strand represents the new DNA that's being formed.
So where do the new nucleotides come from?
And that's a good question. So they come from the food that you eat. It breaks down the chemicals from what you eat and builds up nucleotides. They're then floating around in your cells. And then they can be pulled in. When cells die, they're broken down and recycled. And the parts that make up the cell, like the DNA, the protein, is then broken down to build up new nucleotides that can then be used again to make new cells.
Is it possible for there to be not enough nucleotides to replicate it? And only
Yeah, you'd be malnourished. That would be if you were wasting away and you're very, very thin and you didn't have the right nutrients in your body, no muscle tone. So you would be very, very weak. And yeah, so it's definitely possible, but you would be very, very weak and tired and not have food in your diet if that were to happen. So enzymes and other proteins do the actual work of replication. So enzymes are a special kind of protein that make things go faster in the body. So as I mentioned, one enzyme breaks down the double helix and unzips it. And that's what we see happening right here. And so I mentioned there's nucleotides just floating around in the cells. These come from older cells that have already broken down and from the food that you eat, and they're just pulled in. And you can see right here, that's making the new strand. So the blue is the old strand, and then this gold is the new strand that's being created based off one side of the DNA. And by doing this, so this blue strand over here is a mirror image of this one. So if you see a G on this side, you're going to see a C over here. If you see a T right here, you're going to see an A over here. Same thing right here. The gold strand is just a mirror image of this one, which basically you're copying this strand right here. So by mirroring this side, it is really building the same strand that you're seeing right here. Shall I get these down? DNA polymerase is an enzyme that bonds the nucleotides together to form a new double helix. So as you can see right here, it looks like this blob right here, or right here. So you can see it's building the gold strand right here. It's making the new DNA strand. And you see that there's two working at the same time. One is going to the right, one is going to the left. And you can see that they're now making two new strands of DNA. You have two double helixes that have formed. This one's forming this way, that one's forming that way. So at the end, you're going to have a pair of DNA strands copied exactly. Polymerase enzymes form covalent bonds between nucleotides in the strand. So you have a covalent bond that forms right here that makes a gold ribbon, which is very strong. And the hydrogen bonding that forms between the two nucleotides right here, which is a weaker bond. Did you get those down? So this whole process right here takes 8 hours to go down the 6 feet length of DNA in the cell. And the reason why it can do it in 8 hours instead of longer is that there's several of these polymerases going down the strand at different times to build up parts of the DNA strand. There's 100 or so of them. So two new strands of DNA are formed. You can see this two right here. Each one has an original strand, which you can see in blue, and a new strand, which is gold. So this is called semi-conservative replication, where you have an old and a new strand.
So if you only have two strands in your entire body that are original.
That's right. Yeah. Yeah. So each one, so you have an old one, which builds off a new one. And so when a new cell is formed, the new one had an old strand along with a new strand in it. And each cell has a certain amount of times it can do this before the cell dies. It depends on the kind of cell in your body and how old you are, but anywhere from 30 to 80 to 100 times it can do this and replicate and make new cells. But eventually the DNA wears away to the point where it can't make this happen, and that cell then dies. And that's what we call as aging. The DNA unravels at the ends as you get older. And eventually you just can't replicate your cells. And that's what you see as aging in the person. That's what you see as wrinkles, as, you know, as grayer skin, as being more frail. The body cannot replace the dead cells. When you're growing, you're making new cells at a faster rate than the cells that are dying. If you're not aging, then the amount of cells that die equal the amount of cells that are being made. So in your 20s, that's what's happening. But then into your 30s and older, then the reverse happens. More cells die than are formed. And that's when you start to age. So if you can find a way to keep the DNA from unraveling and keep on replicating the DNA strand indefinitely, then you would not age. The cells wouldn't ever grow old. And so they're working on a way to do this where they keep the DNA from unraveling. If they can find the answer to that question, then you can live for a lot longer and not age.
Do dead cells that still have the DNA in them, or why is it that?
Yeah, some dead cells have the DNA, but it's not always fully formed, or it can be damaged. But yeah, once the cell dies, the body does start to break it down and recycle the parts it can recycle. They don't just stay there. But yeah, dead cells do have DNA, but as it gets older, as the cell decomposes, the DNA starts to be broken down, and it's not going to be functional anymore. But, you know, dinosaur DNA, they still have bits of it. It's not the full strand of DNA. That's millions of years ago, but you can still find some DNA in the dinosaur fossils. So some is still preserved even millions of years later. It's just not the full set of DNA. Replication, as I mentioned, is very fast, especially thinking about what actually happens in this. 3 billion nucleotides are copied in 8 hours. About 50 nucleotides are copied every second. And at this rate, if you just had one of them working, then it would take 33 days to replicate your whole DNA strand. But you have hundreds of points of replication happening simultaneously, so it takes 8 hours. But scientists do think that they're on the verge of a breakthrough where you could live for 100, 200 years longer than we do live right now. So some scientists believe that in the next decade or two, there would be technology in place where you could live for 200, 300, even 1,000 years. Some scientists even go that far. So whether that's true or not, we'll have to see. But some are very confident that they do understand aging to the point where they could correct it in the next decade or two, to the point where you could have very long life. Of course, there are diseases, accidents that could cause you to live a shorter life, but natural causes, it would be very hard to die from natural causes if they can find the technology, which they think they can find in the next decade, to the point you could live for a lot longer than we live right now. Yes.
How would you be able to do that? Like surgery or something?
Well, that's a good question. I don't know. Most likely it'd be like a vaccine you would take, kind of like that, where you'd have a virus in your body, which would then add genes to your cells, which would then make you live for longer. Most likely that's what it would be, a shot you take, where it would just put DNA into your cells. That would be my guess, but I haven't looked into it very carefully, so I'm not sure exactly what they would be, what that would look like. That would probably be what you would see happening.
So if they did this, would you age slowly?
Yes, you would age very slowly. Yeah, they could find a way to do this. Yeah, aging would be not much. I mean, be very, very minimal at that point, because if you can keep the DNA from unraveling, then you're not going to really age. So what happens is that the DNA strand right here has several polymerases at different points, which are copying in each direction. And because of that, it can copy a lot faster than if you just had one polymerase going down the strand. So these dots represent a DNA polymerase at different points, and it's copying in each direction, making a new strand of DNA. Yes.
Do we have to draw this?
No, you don't need to. Just give you an idea of what you can see in the strand. Also, you have DNA polymerases that look for errors in copying. So it's incredible to think about that. It's copying so many nucleotides every second. Millions and billions are copied every second in your body, but yet not many mistakes happen. And there's a reason for that. One reason is you have DNA polymerases which go along the strand and look for mistakes. And if they find a mistake in copying, it corrects it. Yeah, so if you're supposed to, if A goes to T and your body puts a G in there, then it corrects that and puts in the right nucleotide. Now, if it misses these and it continues on, that could cause cancer. And so if you have cancer, sometimes what happens is the DNA had a mistake in the code and your body didn't catch it. And you now have a mutated cell, which can be cancerous.
But then when it usually not pass through the checkpoint?
That's exactly right. You got 2 checkpoints. Good. Yeah. And so you have 2 places where the body will check to see if it's a good cell. And if it's not, it will keep it from growing and duplicating, and it just dies on its own. But sometimes it passes through both checkpoints. So cancer is relatively rare because your body does have fail safes in place that keep the cells that are bad from growing. But sometimes they do pass through the checkpoints and you do get cancer. But with this proofreading, you only have 1 error in every 1 billion nucleotides. And a lot of times that 1 error is not going to be significant. Usually that cell would just die on its own. It's going to catch the error in the next checkpoint. That cell just dies. You got 1 cell that dies. You don't even notice that. So for the most part, even when it does make a mistake, it's not going to be a problem for you because your body will just isolate the cell, let it die, recycle the cell parts, and start over again. It's only a problem if you have a large amount of your body destroyed. So being in the sun and getting sunburn is dangerous because you have a large amount of your cells that have been damaged from the sun. And with a lot of cells damaged, you have a greater chance of having cancer from that. A lot of times skin cancer is very slow growing cancer. And so you can be exposed to a sunburn when you're a teenager and not have cancer from that sunburn until you're adult, 10, 20, 30 years later. Some people who are younger think, well, I got sunburn, I'm fine. So it's nothing to worry about. But that sunburn could have been what caused cancer when they're in their 30s or 40s or 50s. So you don't always see a correlation between your sunburn and cancer. Lots of times it could take decades for that to pop up.
But the sun only causes skin cancer, right? Unless we can know.
Yeah, that's right. Sun only causes skin cancer. Yeah, that's right. Yeah. So it's just skin cancer you get from the sun. So sunscreen is very important, not just because of cancer, but also sun causes aging. So it causes wrinkles, your skin to change and look worse. So sunscreen is important to protect your cells because if they're damaged either from sunburn or just from natural UV radiation without being burned, it does damage your cells. It makes them age, which causes wrinkles. Skin color can change. Of course, cancer can also develop from that. Yeah.
Why does it take such a long time for the skin cancer to develop?
So the skin cells, when you get skin cancer, this cancer cells don't divide very quickly, and they're not going to spread very quickly. So that's one good thing about skin cancer, as far as the cancers go, is that it doesn't usually spread very quickly. It's easy to catch and isolate and take out of the body before it spreads on to other parts of the body. Now, melanoma is the exception. Melanoma skin cancer is faster moving. That one is more of a problem. It looks like a mole in your body. But other skin cancers like basal cell carcinoma, it looks more like a clear blister. That spreads a lot more slowly. And that can take decades for it to spread to your organs and be a problem for you. So your skin cancer cells don't always spread very quickly. And they kind of just stay in one place for years at a time. So of all the cancer you can get, skin cancer is probably the one you would pick because it's very easy to see. It's on your surface of your skin. It's easy to catch before it spreads. It's easy to remove before it gets to be a problem. Of course, it can is cancer. Is the cancer serious? And cancer can be deadly on your skin as well too. So skin cancer can kill you, but of the cancers, it's one of the least deadly ones if you catch it early.
So can skin cancer be hereditary?
I haven't heard that it is. I mean, if you have pale skin, you are more prone to skin cancer. And so it's in that way, it could be hereditary because, if you pass on pale skin to your children, they are more likely to have cancer of skin cancer. Those are dark skin. But no, I haven't heard that. Ovarian cancer, uterine cancer, prostate cancer, breast cancer are the ones that are passed on to children. But I have not heard that skin cancer is on its own. But in a family of all people who have fair skin, they would more likely have skin cancer because they have fair skin, not because they have a gene for that. All right, so that ends 8.3. I'm going to do a couple of things today. We have time. One thing I want you to do is work on page 224, do 1 through 6. They're going to do a quick lab after you do that on the same page. For those at home on Zoom, I'll email you the questions to work on, and I'll end Zoom right now. Do you have any questions on Zoom before I go ahead and close off?
No, but thank you.
All right, have a good day.
Thank you.